Okay, so chapter 5, we're going to be looking at tissues, or the study of tissues, histology, so we can better understand how organ systems work. So tissues are comprised of cells or groups of cells that are similar in structure and form. So we have 4 major tissue types that make up everything in the body. So you have epithelial tissue or epithelium, connective tissue, muscular or muscle tissue, and nervous or nerve tissue. And at least 2 of these major tissue types will make up each of the organs in our body. So this is just a very general kind of breakdown of the difference between these organ systems. We are going to be focusing, right now we're going to focus on the epithelial tissues and the connective tissues. We will talk about the other 2 tissue types when we get to those appropriate chapters. So we're going to talk about basically the differences between each of the 2, and we will talk about a few of the specific types. Okay, so for it to be an epithelial tissue, it's either going to cover or line any external or internal surfaces of the body, or it can be glandular, which means it's secreting some type of fluid. A lot of times these will actually be both. Okay, the lining tissue will also be secretory. So all epithelial tissues, the cells have what we call polarity. And I know y'all talked about this in lab. So if you remember, you have the basal surface, you have your epithelial cell, so the fact that they have polarity, we're saying that one surface is going to be firmly attached. Okay, and of course you're going to have connective tissue underneath that surface. The other surface is going to be completely free. Okay, if you happen to have cilia or anything, this is where this would be. This is called the apical surface. So with skin, the apical surface would be what you touch. In your mouth, the apical surface would be what touches the food or the liquids that are in your mouth. Epithelial tissues, as I also showed, are composed of closely packed cells. So, let's say we're talking about the skin. If you want something to come in through the skin, through the absorption, you want it to have to use the cell transport processes, passive or active, that we previously talked about. Okay, you do not want things to be able to squeeze between adjacent cells. So we have 2 different things that basically help with this. You have tight junctions, which help keep the cells attached to the basal surface. They also keep the cells very close together, so nothing can squeeze through. And then a lot of epithelial cells are under stress. So think about if you gain a little weight, okay, you want your epithelial cells to stretch, again, without allowing things to come through. That's where the desmosomes would play a role. So the desmosomes are going to be there too, allowing for a little bit of stretch without allowing the cells to tear. Epithelial tissue itself is avascular. So vascular refers to the blood flow in the area. A means without. Innervated means that there's nerve supply. So of course you have sensations, you have receptors that can be activated. So because the epithelial tissue itself is avascular, it must have blood supply right underneath it. Okay, so the nutrients can easily diffuse. So you will always have connective tissue immediately underneath. And the type of connective tissue you will always have is going to be loose or areolar connective tissue. That will always be right underneath the cells. Why you want to make sure that they're getting the proper nutrients that they need, gases, is that we say they have a high rate of regeneration, which basically means they can divide very rapidly. And of course, we're making exact copies. Okay, so the type of cell division we will be talking about, mitosis for the nuclear division, followed by cytokinesis. So whenever we're naming an epithelial tissue, there's basically two questions that you have to ask yourself. How many layers is this tissue? If it's only one, it's going to be a type of simple epithelial tissue. If it's more than one, it doesn't matter if it's 2 or if it's 1,000. Okay, we would refer to that as a stratified epithelial tissue. If it's stratified, you might notice that you have several shapes. I would call these right here simple, or I would call it, cuboidal, where these are going to be flattened. Those are squamous. You always name it based on the cells at the apical surface. Okay, so this type of tissue would be referred to as stratified squamous, which is what makes up our skin. The second question you ask yourself, which I kind of briefly just talked about that, is what shape is the cell? And one of the easy things to look at here is also the shape of the nucleus. So if your nucleus looks squished, flattened, and the cell might look squished or flattened as well, kind of scale-like, then we refer to that as squamous or squamous. If the nucleus is pretty much a perfect circle, kind of equal cytoplasm around the edges, we refer to the cell as cuboidal. And if the cell looks elongated, you can have unequal cytoplasm. You might even see it like this, where it's more in the middle. Okay, but you would still have more cytoplasm above and below than on the sides. Still an elongated cell. So we refer to that as columnar. Now talking about when epithelial tissue is secretory. There's a few different things that we can talk about it or classify these. So if the gland itself is made up of one or more cells, and again it produces a secretion, some type of fluid. So we can classify glands based on where the secretion is released. So we're going to talk about the difference between endocrine versus exocrine, and also the number of cells that form the gland. Most of the glands in the body are going to be multicellular. There's really only one important unicellular gland, and that's going to be the goblet cell. So the difference between endocrine and exocrine. An endocrine gland does not have a duct. So what that means is these glands are going to rely heavily on mainly the blood or the lymphatic systems, your cardiovascular system, to basically get the secretion where it needs to go. So a gland with a duct, which is called an exocrine, and most glands as well as ducts typically are going to be simple cuboidal. So again, this is going to be an exocrine type. So when the secretion is produced, okay, it has a pathway to take it to the surface. With endocrine, you do not have a duct. So here, the secretion made by the cells is actually going to be dumped outside of the cells. Sometimes it might be in the lumen and then move to the outside. So remember, the fluid outside of a cell we refer to as the extracellular fluid or interstitial fluid. And typically you're going to have capillaries or lymphatic vessels nearby that secretion will move into, and then it can be carried to its target organ, the organ in which the secretion will carry out whatever job it is. So secretions may include amino acids. If these amino acids are in a long polypeptide chain, it could be a protein. Sometimes these proteins have sugars attached. That's what we call a glycoprotein, as well as steroids. A lot of these, sometimes if it's a hormone, it's typically going to be a steroid or a protein. So we go back to this drawing. This is going to be the duct portion. Okay, this is just carrying the secretion to the surface versus the cells that actually make the secretion would be, of course, the secretory unit. As we said, exocrine have ducts, and in the body, most of our glands tend to be exocrine. Those, of course, that are part of the endocrine will always be hormones. As I mentioned, typically they're made of simple cuboidal epithelium. If it's not simple cuboidal, then it's typically columnar. The secretions will be released onto body surfaces. So with your skin, you would have your sweat glands or the oil glands, or into body cavities. So like with your stomach, the secretions, the acid, the other fluids would be in the lumen of your stomach, and that would mix with the food that you're eating. So examples, again, in the skin, you have sweat and oil. Mucus would be in the respiratory tract. Salivary glands making saliva to help moisten your food before you swallow. There are several examples. And again, most glands are multicellular, so the multicellular portion will be composed of a duct as well as a secretory unit. So the important unicellular cell is what we call a goblet cell. And basically, its Golgi is going to make these little vesicles filled with mucin. And this mucin is exocytosed at the apical surface of these cells. And of course, one of the main components of extracellular fluid is water. So when the mucin mixes with water, it becomes mucus. We, of course, find goblet cells in the respiratory tract between the pseudostratified ciliated columnar cells, as well as between simple columnar cells within the digestive tract, stomach, small or large intestines. So we can classify glands based on how the secretions get to the surface of the cell. So if it's called merocrine, or you can actually sometimes see this as eccrine, then the products are secreted by exocytosis. Apocrine, the products are lost as a result of the apical cytoplasm and the secretory product. This one, and examples of these in the body would be in the pancreas, secreting the digestive enzymes, sweat glands in the skin, as well as salivary glands. Apocrine release their contents from the apical cytoplasm. So basically, mammary glands giving milk, secreting milk in the breast are an example of this type. Holocrine secrete by rupturing the entire gland. So basically the cell is going to fill with the secretion. Sebaceous or oil glands work in this way. So if we're looking at this here, the Golgi apparatus is the main organelle which packages things that are going to be transported within the cell or out of the cell. So it makes the vesicles, they go to the top of the plasma membrane, they're going to fuse with it, and then the contents are released. So that will be a type of merocrine gland. You're going to see that the secretions are in vesicles, and it's going to fill the apical portion of that cell. And once the apical portion is all filled with the contents, it's going to start to pinch. And basically, it releases this whole part of the cell. The cellular components break down, and then the secretions that are in the vesicles are then released. The entire cell. Okay, then you're just going to basically rupture the plasma membrane, so it's similar at that point, and then the secretions will just be released at the apical surface. Merocrine. Okay, so now moving on to connective tissues. It's the most abundant and widely distributed tissues in the body. It basically fills in between the other tissue types, between your epithelial tissues and muscle, as well as a lot of the actual other connective tissues as well. It has a very wide range. It can be anything from bone to blood. All connective tissue is going to be derived from the embryonic connective tissue, which we refer to as mesenchyme. So as the embryo continues to grow and develop, the mesenchyme will start to differentiate into the 3 different classes of connective tissue that we still see in the adult. So you have connective tissue proper. These are mainly made of fibers that might either be loosely or densely packed. Fluid connective tissues mainly are going to be the blood and lymphatic. And supporting connective tissues, you want to think about skeletal tissues. So this is a nice kind of flow chart that separates the specific type of connective tissues. You do need to know where each of these types of tissues could be located in the body, as well as with the epithelial tissues. Refer to page 58 and that table you made with the tissue type and the locations of their body to review those for your exam. You will not have to be able to identify them based on picture, but based on their function or where they're found, you will have to know that. So functions of connective tissue, again, can be very diverse. Binding would include your dense regular connective tissue, which makes up tendons and ligaments. Tendons will connect muscle to bone. Ligaments connect bone to bone. Supporting tissues, these could include a lot of your other connective tissue propers, areolar, anything that kind of packages or surrounds other organs. For protection, you have a few different ones that can be seen this way. You can look at bone as protection. The most protected organs in the body are going to be the brain and the spinal cord. Cartilage can also help protect in that it absorbs some shock. You have insulation, of course, which would be the fat. So with insulation, that actually does protect deeper tissues because fat can absorb some mechanical or physical stress, as well as it's going to help maintain your body temperature. And then transportation would be movement of possible gases and nutrients or metabolic waste to and from other types of cells, which again, this would be primarily done by your cardiovascular system, the blood, but the lymphatic system, lymph vessels, the fluid they contain is called lymph. It's very similar to interstitial fluid. It can carry some as well. So just like we looked at the common characteristics between all epithelial tissues, we're going to do the same thing for connective tissues. Again, as I previously mentioned, they're all going to come from mesenchyme. So before it becomes any of the specific types of connective tissues, it was mesenchyme. They do have what we call varying degree of vascularity. Most connective tissues have blood vessels. We will talk about a few that do not. They're all going to have living cells. Sorry, this part's living. Cells are living. The non-living part is the extracellular matrix, which is made by the cells. So that's going to include what we call ground substance and fibers. So what ground substance is? It's the unstructured material that fills the space between cells. So basically you're talking about interstitial fluid. Okay, but it has some unique characteristics and some of the things that can be stored here. It can be very watery, more like interstitial fluid, or the ground substance can be really thick and more gel-like. There are 3 different fibers that can be found within connective tissue, and we're going to discuss the differences between those. And then most of the time in connective tissue, you have very specific types of cells. So chondro is going to refer to cartilage, so those are going to be found in cartilage. Osteo are found in bone. So basically when we get to each connective tissue in the body, we will discuss that. We'll say fibroblasts are found in lots of different types of connective tissue. Most of those in the connective tissue proper. A blast is a cell that builds. So basically fibroblasts are going to lay down the different fiber types. Just by knowing the type of fiber that's found in the connective tissue, that will give you a good idea of what its possible functions could include. So there's 3 different fiber types that can be present. A tissue can have all 3, it can have 2, it can just have 1. And again, remember, these are connective tissues. So collagen fibers are known for their strength. They provide, we call, a high tensile strength. When they're stretched or you try to break them, it's very difficult, okay, for them to break and tear. Commonly, they're called white fibers. They do appear white in a cadaver. Elastic fibers are going to be thinner, and tissues, they tend to appear kind of wiry, very, very thin, and these allow for stretch. Think like a rubber band. They allow you to pull and stretch it out, and then they will recoil or return to the original shape. And then reticular are a specific type of collagen fibers that are highly branching. They basically form these little branching meshworks that typically will form a type of matrix. And really, one of the more common places we find reticular fibers is in reticular tissue, which is going to be found in lymphatic organs and tissues. So again, make sure that on page 58 in your lab manual that you go over that chart that you filled out and you know the locations and general functions of the types of epithelial as well as connective tissues. Body membranes. There's 4 different types we want to talk about, 3 of which are comprised of epithelial tissue. Only 1 is with connective tissue, and we'll actually discuss the synovial further when we get to joints, which is after the skeleton. So differentiating between these types of membranes, one of the types of epithelial membranes is called the cutaneous membrane. Cutaneous refers to skin, and specifically the skin is comprised of stratified squamous epithelium. So again, stratified layers are going to give it function for protection. Then you have the mucous membranes. These are going to line body cavities, organs that are open to the exterior. So in the picture, they're showing the respiratory tract, which technically you can breathe through the mouth as well, but it's going to come here anterior, of course, go to the lungs, as well as your digestive tract, stomach, and it would go all the way through the intestines. They're just not showing that. Also, what's open to the exterior would be urinary or urogenital tracts, the reproductive and urinary systems as well. These tissues typically have a fluid over it. With digestive and respiratory, the small amount of fluid is produced by the goblet cells. Okay, so digestive, starting at the stomach, you're going to have simple columnar with interspersed goblet cells. You'll have the same thing in the small and large intestines. Respiratory tract, starting at the nasal cavity, your larynx, the trachea, the bronchi, those are all going to be your ciliated, pseudostratified columnar epithelium. And the last type of epithelial membrane we've actually discussed before. Y'all really discussed it in lab. So that's serous membranes. Serous membranes are very thin, simple squamous or squamous epithelium, and they produce a very small amount of fluid. And basically the purpose of the fluid as well as the membranes is to reduce friction. When you look at the torso models, you'll notice that all the organs are tightly packed within the ventral body cavity. So those are the cavities where we will find the serous membranes. As your heart expands, which is medial to your lungs, the lungs expand. Okay, as you eat too much in your stomach, you don't want any of those organs creating friction and therefore damage, putting pressure on other organs that surround them. Okay, so these serous membranes help to prevent that. So if it is a membrane that is lining a body cavity, we refer to it as parietal. If it directly covers an organ, we refer to it as visceral. and then you can use each of the specific names depending on what body cavity you're talking about. So of course the lungs, we're going to have pleura around the heart, pericardium, and then any of the organs within the abdominal or pelvic cavity, we're going to use the term peritoneum. So now that we've briefly discussed the epithelial and connective tissues, we're going to talk about how tissues repair themselves. And basically, when you first injure a tissue, what are some of the steps involved with trying to return that tissue to the original healthy tissue? So there's two major phases for this. The first phase is part of the injury. These are a lot of the symptoms that you feel, okay, right after you've hurt yourself. We're going to talk about what all is involved with inflammation. And then basically, once you kind of clear out the damaged stuff, now we can actually start making the new tissue. And that's going to be part of the repair phase, which is going to have two steps to it: organization and regeneration and fibrosis. So first, we are going to discuss inflammation. So as soon as you injure yourself, your tissues start to release chemicals. They are inflammatory chemicals. These inflammatory chemicals have what we call a chemotactic that actually might be an X. I'm sorry, I'm not the best speller. Chemotactic nature to them, which basically means it's like they put off an alarm, and that tells other things, specifically here, other white blood cells, that they need to come to that area. One of the types of white blood cells that's going to come to the area are going to be what we call, once they enter the tissue, we call them mast cells. And these mast cells have granules in them. And the granules have two different chemicals. Both start with an H. So histamine, which is a type of inflammatory chemical, as well as heparin. Heparin is a blood thinner or anticoagulant. So once this chemotactic alarm goes off, you're going to have a lot of white blood cells coming to the area. You don't want them to accidentally form a clot and possibly block the blood flow further down. So that's why you release a small amount of heparin. And I should be specific and say this is all localized. Okay, so you're only releasing these chemicals at the site of injury. So the histamine, as well as the heparin, they're going to do things to the blood vessels in that area. So they're going to increase the amount of blood flow. They do this by dilating the blood vessel. So the smooth muscle within the blood vessel relaxes. That allows more blood to come to the injured area. And then also the vessel wall is actually going to become more permeable, which means it's easier for nutrients, gases, white blood cells to leave the blood and enter the injured tissue. Also, most of the time when we injure ourselves, you've broken some blood vessels. So you want to clot to stop the bleeding from spreading and losing too much blood within your cardiovascular system. So these are what we call the 4 cardinal signs of inflammation. These are symptoms that you will feel or a patient might describe, and you know that basically the first part of the healing immediately after injury has begun. So because you have more blood in the area, that's why you're going to have swelling. So you literally have more fluid. You also have more fluid that's entering the tissue, remember, because it's more permeable. So both of those create swelling in the area. Because of hemoglobin found in blood, blood has a red color to it. So because you have more blood in the area, you're going to have more of a pink-red tint in the area that you've injured. Blood's also slightly warmer than the rest of your body. It's about 100.3 degrees Fahrenheit. So you're going to feel warm in that area. The swelling will cause some pain. Typically, though, when you've injured yourself, you've also, with the release of these inflammatory chemicals, they bind to pain receptors. So you feel pain. Okay, so here we're basically showing someone has, obviously cut through their skin and they've cut through the blood vessels. So the blood vessel's leaking. Okay, so the first thing that's going to happen is you basically have to imagine all of this that you cut through, that tissue is damaged. We want to get rid of it. Okay, so it's going to become necrotic. It's going to die. How does it do that? The cells go through apoptosis or autolysis. So they're going to release the enzymes within the lysosomes, and that will basically degrade all of that portion. So your dead cells create pus. Also, pus can include some bacteria that may have entered the body because you have made that cut or injury as well. Typically, the body will either push the bacteria and dead cells out, or a lot of times it will actually reabsorb it and get rid of it within the bloodstream, majorly by these type of cells, which I'll discuss in a minute. If that doesn't happen, okay, and your dead cells and possibly bacteria stay kind of trapped underneath the surface and not in the blood, then you can get an abscess, and that must be lanced, and you must allow those cells and the bacteria to leave. So immediately when you have bleeding that occurs here, you're going to start to form a clot, okay, to stop that blood flow. And basically your scab is that old clot. Okay, so the blood clot's going to start to form, and you will barely start to see some scabbing there. So you have these damaged cells that have died because of the enzymes that they've released. Again, possibly microorganisms like bacteria that shouldn't be there. So we want to avoid an infection. So that's the major job of the macrophages. They are phagocytic. They engulf and break down cellular debris or microorganisms that should not be there. And typically the body is able to avoid infection, hopefully, by this process. So now we've had blood clotting, we've stopped the bleeding, the blood vessel was damaged, so we removed those damaged parts. So now the ends of the blood vessels basically have growth factors, okay, that start to be produced, and that basically makes it start dividing, and it's going to start growing to form, okay, the complete blood vessel back together. You see that the scab, okay, so your clot that becomes the scab, that's getting pushed up by this newly forming epithelium. So because it's undergoing cell division very rapidly, that's basically what we call regeneration. You also have fibroblasts here. These fibroblasts are going to start to produce collagen fibers. Again, you have the white blood cells, the macrophages that are going to continue to phagocytize anything that shouldn't be there. Okay, so slowly this regenerating new tissue is going to keep going up, and it will push off the scab. Why you don't want to pick scabs off early. If you do too early, you may not have enough tissue here, and so you may allow the area to bleed again. It should be a T. Okay, so now your new tissue's formed. Okay, your scab could be removed, and the fibroblasts basically are going to really start laying down the collagen fibers. So as they lay down the collagen fibers here, that's going to become your scar tissue. And the idea is that eventually we want the newly formed tissue to look like what it does around it. Okay, so we want it to be completely formed. Again, here you would have the fibrosed area, okay, which is basically your fibroblast again, because the growth factors lay down lots of collagen fibers. This may make the area be slightly raised for a little while, but typically through long-term processes, macrophages may come and get rid of some of that extra scar tissue. We will discuss that a little bit further when we do the integumentary system. Okay, so we're talking about tissues that can regenerate quickly, basically divide, replace themselves. So highly regenerative means they have a good capability of making new tissue rapidly. Epithelial tissue is really one of the fastest regenerating tissues in the body. Hematopoietic tissue, that's what red bone marrow is. And that's where actually all blood cells are produced. The type of connective tissue, loose areolar, which is always found underneath epithelial. It's highly regenerative as well. And then bone. Moderate, so it still regenerates pretty good, pretty well. We have smooth muscle, which forms all of your hollow organs. Dense regular tissue, which is going to form ligaments and tendons. Ligaments connect bone to bone. Tendons connect muscle to bone. And dense irregular tissue, which makes up joint capsules, and it's also found within part of the skin. So think about if you sprain or strain your ankle, you've basically injured a ligament or tendon. And a lot of times that will take longer to recover than if you have a simple break of a bone. Why? Because bone has a higher rate of regeneration. And the major reason why bone has a higher rate of regeneration than dense regular is because it's well vascularized. So we saw with inflammation the importance of the blood vessels. That's how the mast cells and the macrophages get to the area that they need to go. So the more blood vessels are in the area, that's really going to play an important role with the speed of recovery as well. One of the reasons why cartilage is under weak. Cartilage is one of the few types of connective tissue which is avascular, which means it does not contain blood vessels. That's why if you pierce your ear, you leave it in for a long period of time, it will not heal back. You'll see the same thing with belly button. If you look at fighters, wrestlers, mixed martial artists, MMA, they get what they call cauliflower ear. The ear gets this whole funky, big built up mass, and that's because the cartilage is getting busted and it doesn't heal properly, because although the lining around the cartilage, the perichondrium, is vascular, the cartilage itself is not. So the healing rate is not very good. Skeletal muscle is well vascularized, but it takes longer to recover. And a lot of skeletal muscle recovery also has to deal with physical therapy. No regeneration, we're basically talking about when these tissues are mature. Okay, so once your nervous tissue is mature and the cardiac muscle, which forms the heart, is mature, if these tissues get damaged, they cannot regenerate. So once the cells die, they stay dead. That tissue stays necrotic. That's how you can actually determine how serious a stroke is. Okay, so a stroke is when you have blood impairment to the brain or to the heart. And so both of those cases, if too many of the cells within the nervous tissue die or too many cardiac muscle cells die, the person cannot survive the incident. Okay, if it's a minor heart attack, there's enough living tissue to still provide an adequate pump for the cardiovascular system. If the stroke is minor, or even just half the side of the brain or small portion of the brain, you can actually train neurons to do new jobs. So if you're like, someone's had a stroke and they learn to talk and stuff again, that's not because new neurons are forming. It's old neurons are learning to do new tricks, essentially. So now we have stem cells. Stem cells are cells that are not fully differentiated, so they're not mature. So we can use stem cells to repair or replace damaged or dying tissue. So for a cell to be considered a stem cell, that means it exhibits self-renewal, so it can at least replace itself, and what we call potency. The potency is how many other cells can it become. Basically, we're asking how far differentiated is the cell. So if a stem cell is referred to as totipotent, That means that the cell can become any cell in the body. Okay, it's limitless. So you only find these in very early stages within the embryo. Pluripotent stem cells are also only found in the embryo. It's a little bit further on, and basically they can form any of the tissue germ layers of an embryo, so still very undifferentiated. Multipotent, we do see in adults, and of course we also see this in the embryo again. In adults, it's partially differentiated. That's what we have in the stem cells of the bone marrow. So essentially in the bone marrow, you have one cell that can become a red blood cell. It can also become several different types of white blood cells. That's why it's referred to as multipotent. So again, that's found in our red bone marrow. In the embryo, so this would be further along in development, once these cells become mesenchymal, they can still become any type of connective tissue. Okay, so that would be considered multipotent as well. Unipotent, we only see in adults, and basically they can only differentiate into one type of cell. Basically, we're saying they have the ability of self-renewal. So most epithelial cells are highly regenerative. They can replace themselves. So there's a lot of controversy about stem cells. So how do we collect these stem cells? Most are collected, harvested from what we call a blastocyst. So basically, as soon as your mom, she would have the sperm, which has 23 chromosomes, we call haploid, and that's going to fertilize the egg. Okay, which has 23 chromosomes, also haploid. So we're going to add those together. That becomes what we call the diploid number, which means 46 chromosomes, which is how many chromosomes all of our cells have except the sex cells. Now that is your zygote. And this zygote's going to start rapidly dividing. Okay, and this little ball of cells, this becomes what we're going to call the blastocyst. So the blastocyst is going to contain those totipotent stem cells. Further along, this is actually what's going to become the embryo. So this ball is going to hollow out. These cells could then also become the pluripotent stem cells. So is this ethical? That's the question that people have. So basically, how do we end up with embryos that might possibly be in these different stages of development? Okay, why would you have that? Basically, the main reason for that would be for couples that cannot conceive naturally. So they can still, the woman still has eggs that are collected, sperm are collected from the male. They basically do this process in a test tube and they allow it to start dividing. And then they're going to insert the cell back in the female and hope that it attaches to the uterine wall. Many times when they do this, it's called in vitro fertilization. There's a lot of hormones, procedural things that the female and possibly male might be going through. So a lot of times they will collect several of these different blastocysts and store them, possibly freeze them to give the couple several opportunities, to try to conceive a child. And so if a couple is lucky, okay, maybe they have 3 other embryos that are frozen that they're not going to be used, then possibly those could be used for research purposes. What we also can do, and this is why you have the choice of collecting cord blood, that would be blood from your umbilical cord. That can be saved and used later for the individual for stem cells that are available. Those typically will be multipotent, possibly even pluripotent, or possibly even from someone in the family if they have similar enough tissue types. Adult stem cells, most of the time we're talking about taking them from the bone marrow. So that's why if someone has a type of cancer of one of the blood cells, you can remove all of their bone marrow, take healthy bone marrow from another person, insert it into the hollowed out, and you allow those cells to divide and basically create the new population. So as the embryo is growing, we were talking about earlier, it's in this hollowed out area. Basically, it's going to differentiate into 3 different germ layers. So you have the ectoderm, and sorry, this text is going a little over. The ectoderm is going to be the blue here. Ectoderm is going to form all nervous tissue. The mesoderm, that's the red, it makes up kind of the meat of your organs. That's going to be comprised of muscle and connective tissue. Will all come from mesoderm. And then epithelial tissue or epithelium can actually come from all 3 different germ layers. And what's kind of interesting here, and you don't have to know this testing over it, but as this disc forms, it will actually then fold upon itself. And so it's kind of interesting here. This is called the neural groove. That will become the spinal cord. This will become the brain. And this is an embryo at 16 days. Okay, so those very rudimentary forms of those structures are already starting to form. So as tissues develop and as we age, a lot of times they go through changes or modifications. Okay, so you need to understand what the difference of these terms are. So a lot of these are going to end in the term plasia for changing, okay, modifying. So metaplasia, you have mature epithelium that changes to a different form. Usually it's to help the person out. It's to benefit the person, help adapt to their environment. So smokers, the pseudostratified ciliated columnar epithelium constantly is getting irritated, okay, by the smoking and what they're inhaling. And even though it looks like multiple layers, keep in mind that's why we call it pseudo-stratified. It's falsely stratified. It's really only 1 layer. So over time, they're going to want to thicken up that tissue to be more protective. So it's going to become what we call non-keratinized stratified squamous. That's the same type that would be found in your esophagus and your mouth. People that have chronic acid reflux, so chronic means long term. Typically you're talking about 2+ weeks. Esophagus is non-keratinized stratified squamous, just what we talked about earlier, okay, what the trachea might become. So it's going to develop more like the epithelium that we actually have in the stomach. The stomach is where the strong acid, hydrochloric acid, is produced. So the stomach has goblet cells, which form a lot of mucus, which help line and protect the actual simple columnar epithelial cells of the stomach. So basically you're going to develop the same thing in the esophagus. So you're going to have the simple columnar with the goblet cells to help protect the esophagus from the strong acid. Okay, hypertrophy and hyperplasia. So both of these, you're talking about an increase. Trophy, the tissue itself isn't changing. The cell in the tissue is increasing in size. Okay, so if your cell used to be this size, now it's going to be this size. And if all of the cells in a tissue are enlarging like that, then yes, the entire tissue would get larger. Hyperplasia, you increase the tissue as well, but you do it by increasing how many cells you have. So the cells themselves are not getting larger, you're increasing in number. We will talk about when the body does each of these when we discuss a little bit more about the skeletal and muscular systems. Neoplasia is basically division, growth that is uncontrolled. It develops or divides too much, so it can develop into a tumor of the abnormal tissue, typically a cyst that typically will have to be removed. It may or may not, we're not necessarily saying it's cancerous at this point. And then as we get older, one of the major things, and this is actually how we see aging in people, is that we have atrophy of tissues. So tissues decrease in the number of cells and in the size. So both of those are going to drastically make, for example, less skeletal muscle. So you lose strength. Your skin thins. And if too much of this occurs, it actually might make the organ not be able to maintain homeostasis anymore. And you could have organ failure, which of course could lead to death. And individuals that cannot move around, the muscles that they're not using, they're not being used, so your body doesn't need to maintain them. So they basically start to eat themselves away and you lose all strength. They become smaller and smaller. And with physical therapy, at least some of that can be reversed. You may not be able to get all the muscle as you did before, but at least some as you do strength exercises. And then talked about this earlier, necrosis is when the tissue dies. This is due to the tissue being damaged. So basically the cells are damaged, they go through autolysis or apoptosis, and that tissue becomes necrotic. We talked about this earlier with inflammation. So that's the first part of the response once you've had an injury. So again, that injured tissue, we want to get rid of it. If the body can't absorb these tissues, basically you have rotting tissue, which we call gangrene, and you have an infection that if you don't remove that limb, remove the gangrene wherever it is, that it will get into the blood, spread through the whole body, and cause death. So as we age, because of the atrophy, and because our cells do not basically divide as often, our cells are not as efficient. So again, as I mentioned earlier, your epithelial cells aren't dividing at as fast a rate, so your skin becomes thinner. Connective tissues don't have as many collagen fibers, elastic fibers, so you lose the stretchiness, the recoiling ability, the resiliency, the strength in your skin. You also see that in ligaments and tendons. So talked about this collagen. Declining collagen is one of the reasons why you do have less flexibility. You do have sagging of the skin. Hair might become more brittle. Your tissues take longer to repair. Bones are not replacing their tissue as often, so they become weaker and more brittle. Muscle and nervous tissue begins to diminish if you don't use it. That's why it's important to do exercise and keep your brain active. And you can try to put off aging as much as you can by having a healthy diet and being highly active. So people that have a poor diet do not move around very often, which affects circulation, or might just have circulatory problems. They're going to see aging of tissues much more rapidly.